one is like intercalating dyes and one is probe base. So what are the terminologies when you perform qPCR, and you will encounter that, so you will get a good clarity when we finish this lecture. And at the end we will give more focus on how we can perform virus load estimation using qPCR. So I think you all have taken PCR lecture. Okay, so what happens in first step? So I am doing, this is denaturation, right? The hydrogen bond breaks because we raise the block temperature. And how much the temperature? 95. If I raise to 98, then what will happen? In some cases we used to raise it for 98. It depends on the ability of a enzyme to withstand that temperature. But we don't raise it for like 4 degree, 5 degree, 4 minutes, 5 minutes. We just raise it for 30 seconds. Then what is the next step? Annealing. And what happens in that? Primers will bind, right? So which primers will bind to which strand? Forward will bind to which strand? Sense or anti-sense? Sorry? I can't hear. Which strand? Like this one is the leading strand or sense strand. This is the anti-sense strand. Forward primers will bind to sense or anti-sense? Anti-sense, right? So let's see. Forward will bind to the anti-sense and reverse will bind to the sense. है ना? Then what is the next step? Extension. It is doing, right? But wait a minute. You can see here our forward primer starts here and reverse primer ends here, right? So we must get a PCR product within the range of forward primers at the end of the exactly same at the end of the reverse primer. But when this strand is synthesized, there is no one to stop it here. Who is stopping? No one. So it will keep on synthesizing. It depends on the ability of the enzyme, how much it will synthesize, what is the speed of the enzyme, and how much time you are giving for these reactions. So we are not getting specific product. Then how we are getting specific bands PCR में? कोई बताएगा? Anyone? ऐसा ही होना चाहिए. This is happening, right? So what will happen in next step? थोड़ा example लेते हैं. Next step में क्या होगा? Again, वो पहले का example ले रहा हूँ. Again we will denature it. Now this newly synthesized strand in last cycle, now you can see it will act as a antisense. And antisense को कौन सा primer bind करेगा? Forward primer, right? So forward primer के end पे 3' पे enzyme आके बैठेगा. Now it will start synthesizing it. Now you can see in the antisense it can't synthesize beyond the end of the reverse primer जो पहली cycle में synthesize हुआ था. Clear है? तो ऐसे each product will go. Now last time जो synthesize हुआ था product, when we denature it, now this is specific product what we are expecting, right? So in normal PCR this happens and we keep on going for 35 cycles, and in each cycles the PCR product get doubled, and at the end what we see a specific product on the gel, right? That is measured based on the size of the ladder, right? Then how are you sure this is 100% our desired product? Are you sure 100%? We are not sure still, है ना? वो हम लोगों को एक बार तो सीक्वेंस करके देखना पड़ेगा, है ना? First time when you are checking the specificity of the primers, you take it out, purify it and go for sequencing. Then we are 100% sure if it is matching with the हमारा target gene के साथ match कर रहा है. So this is about basic about PCR, है ना? थोड़ा सा clarity के लिए. Now we go for qPCR. What happens in qPCR? The same things will happen, but we are able to measure the accumulated PCR product at the end of each cycles because of fluorescent reporter molecules. They are called as reporter molecules because they report the fluorescence to us or to the machine, है ना? So because this accumulation of fluorescent product is directly proportionate to the PCR product, so हम लोग उसको measure करेंगे तो PCR product भी measure हो जाएगा automatically, right? So there are two chemistries which are used in qPCR. First is the intercalating dyes and second is the probe base. Intercalating dyes most commonly used is the cyber green. वो जब cyber green standardize भी नहीं हुआ था उससे पहले हम लोग ETP है जो एथिडियम ब्रोमाइड है उसको भी ट्राई किया था उसको यूज करने के लिए, बिकॉज वो क्या करता है, दैट इज ऑल्सो इंटरकैलेटिंग डाई. हम लोग विजुअली देख पाते हैं ना, व्हेन वी सी इन यूवी लाइट आप डीएनए को देख पा रहे हैं बिकॉज इट इज इंटरकैलेटिंग डाई. इट गो सिट इनसाइड द डीएनए एंड इट गिव फ्लोरेसेंस व्हेन यू एक्साइट विद यूवी लाइट, है ना. तो सेम लाइक साइबर ग्रीन, इट इज सेफ वन, वो आप इसको यूज कर सकते हैं ना. सो बोथ हैविंग प्रोस एंड कॉन्स एंड वी विल सी वन बाय वन इन डिटेल. तो बोथ इज हैविंग pros and cons. Intercalating dye, it has a very limitation is that because it is not specific. Wherever it will find a double stranded DNA, it will go and sit there and it will give fluorescence. But it doesn't happen in tag probes. These are very specific and very sensitive. So तभी हम लोग प्रेजेंस एंड एब्सेंस के केस में हम लोग वी कैन यूज टैगमैन ओनली. लाइक कोविड के केस में वी वर यूजिंग टैगमैन प्रोब्स ओनली बिकॉज दे आर वेरी वेरी स्पेसिफिक. इफ दे डोंट माइंड दे डोंट गिव फ्लोरेसेंस. So let's see cyber green, what happens in cyber green. In cyber green, let's say this is my first cycle and second cycle the PCR product get doubled. Now the floating cyber green go and sit inside the PCR product. When you excite with light, it give fluorescence and machine will record it, है ना. So whatever recorded fluorescence, it will be shown as a graph. ठीक है. So what happens in next cycle, my PCR product get doubled, so the fluorescence also get doubled. Now it will be recorded as a graph. So the fluorescence is proportionate to the PCR product accumulated at the end of each cycle. This happens, same happens in the next cycle. So again it will be recorded, right? So at the end you will be seeing graphs like this on qPCR. So now you can see wo graphs when they are exponentially growing, mean at the end of each cycles when PCR product is doubling, you can see like straight graph. When the PCR consumables get exhausted, you will see a plateau and a linear amplification. So when you are using a cyber green chemistry, you can use only for quantification, not for like presence absence and SNP genotyping. So quantifications, there are various ways. We will be discussing today in detail about the standard curve, absolute quantification by generating a standard curve. Second is a relative standard curve. It is a mixture of both one. Here we will generate a standard curve. Also we use a endogenous control or a reference gene in comparative city or data which is used for gene expression studies. So when you perform a qPCR using cyber green chemistry, we have talked that it is not specific. To check whether it is specific or not, we perform a melt curve analysis after finishing the qPCR. So what happens when you gradually raise the block temperature from 70 degree to 95 degree, there will be some point where all PCR product will break, they will get separated. So whatever dye was there inside the double stranded DNA, it is released suddenly and it give a peak. So if it is a only single PCR product, we get a single peak. If it is double, mean two, one is non-specific, one may be specific, we don't know, we have to confirm by sequencing. So it is not desirable, this one. और अगर ऐसे ऐसे ग्राफ भी आता है वो भी डिजायरेबल नहीं है. क्लियर है. Another one is tagman chemistry. So we don't use any intercalating dye. Instead we use a Tagman probe. This is the Tagman probe, which is unique than the primers. It is unique than the primers in two aspects. One is it is lacking a free hydroxyl group. So our tag polymerase cannot synthesize by sitting on the 3' of this probe. वो इधर बैठ के इसको आगे synthesize नहीं कर सकता. अगर वो synthesize करेगा, then it won't give fluorescence. One, it lacks a free hydroxyl group, so it cannot be synthesized by tag polymerase. Another, it has two molecules attached. One on the 5' that is called as a reporter molecule. On 3' it is called as a quencher molecule. Quencher molecule, when this probe is intact, and when you excite with light, it don't give fluorescence. No fluorescence is generated, because whatever fluorescence is generated by the reporter molecule is neutralized or absorbed by the quencher molecules through a process called as FRET, fluorescence resonance energy transfer. So there will be no net fluorescence. If we separate these molecules, reporter molecule, quencher molecule, permanently, then if you excite with light, it gives fluorescence. So how we can separate it? अगर probe जाके bind करेगा उधर, next cycle we can see, if our forward primer and reverse primer and probe binds there at the specific sequences, what happens in extension? Enzyme will sit on the 3' of the primers and it will synthesize, but it will cleave the probe. So both the molecules like reporter molecule and fluorescence molecule, quencher molecule get separated permanently, and when you excite with light, it gives fluorescence. If our probe don't bind it, if it don't find the complementary sequences, mean it is very specific, in that case there will be no net fluorescence. So we can use for presence and absence study. Clear? So when you are using this chemistry, you can use it apart from quantitations, you can use for genotyping and presence and absence study. Genotyping में you have to use two probes. One is targeting the wild type, one is targeting the mutant type. अगर mutant है तो mutant में fluorescence मिलेगा, अगर wild है तो wild में fluorescence मिलेगा. दोनों probe एक ही reaction में जाएगा. अगर if it is in heterozygous conditions तो दोनों probe fluorescence देगी, है ना? दोनों probe fluorescence देगी तो दोनों का fluorescence measure करेगा, वो अलग अलग wavelength पे होगा, मशीन उसको automatically record करेगी. And presence absence कोविड का example मैं already quote कर चुका हूँ. So अगर आप we are performing qPCR, you will see a terminology called as threshold. It is a horizontal line which is drawn on the y-axis of this fluorescence. It is drawn on the y-axis, which can be moved up and down in the machine. Not here, it is just picture. So it can be moved and up down. We have to keep in this log phase only, because there the data are predictable. अगर इधर करेंगे तो आपको कोई prediction properly नहीं मिलेगा, इधर नीचे जंक में लाएंगे तो भी नहीं मिलेगा properly. So ये horizontal line that is drawn on the y-axis, ये बताएगा हमें कि where to take the data. ठीक है, अभी ये ग्राफ तो मिल गया, इसका क्या करेंगे? We have to take the data, we need some data to compare. So another term is the CT or cycle threshold. Last time we drawn a horizontal line on y-axis which is called threshold. This cycle, these are the cycles, this is the fluorescence. This cycle in which our real signal fluorescence crosses the threshold line is called as cycle threshold. This is also called as CK value or CT value. है ना? So this form the basis of the quantitation in real time PC, CT value or CK value. Let's take the example of these two samples. One is sample A, sample B. Sample B is having more initial template. So initial template ज़्यादा होगा तो what we will get? Less CT or higher CT? Less CT definitely, because you can see here the PCR product accumulated is more than the sample A इन सेम साइकिल, राइट? सो पीसीआर प्रोडक्ट ज़्यादा है तो फ्लोरोसेंस भी ज़्यादा होगा, है ना? तो पीसीआर प्रोडक्ट ज़्यादा है तो फ्लोरोसेंस भी ज़्यादा होगा. सो इट विल जनरेट ए सफिशिएंट फ्लोरोसेंस विच कैन ओवरकम द बैकग्राउंड नॉइज एंड क्रॉसेस द थ्रेशोल्ड. सो अगर टेम्पलेट इनिशियल ज़्यादा है तो अर्ली सिटी होगा, अगर लेट कम है तो लेट होगा. सी इस सैंपल को, सैंपल ए को एक और साइकिल चाहिए टू जनरेट सेम डेटा और सेम फ्लोरोसेंस. सो एक सिटी इसका लेट आएगा उनमें. इसका अगर 12 है तो इसका 13 होगा. इसको एक और साइकिल चाहिए जिससे ये सेम अमाउंट ऑफ ये डीएनए टेम्पलेट जनरेट कर सके. क्लियर है? अनदर इज बेस लाइन. ये ज़्यादा इंपॉर्टेंट नहीं है बट मैं थोड़ा वैसे ही बता देता हूँ. You can see like this is the junk like things graphs here na. It also have some impact on our real signal. If you don't set the baseline you will see peak like this, है ना? All peaks are not starting from point 0. If you reduce this background noise, वो उस सैंपल का ही बैकग्राउंड नॉइज रिड्यूस करना है उसके ट्रू सिग्नल से. सैंपल ए का बैकग्राउंड नॉइज को ही सैंपल ए के ट्रू सिग्नल से डिटेक्ट करना है. वो सॉफ्टवेयर विल डू इट ऑटोमेटिकली. समटाइम इट फेल्स. इस एग्जांपल से यू कैन सी हियर, राइट? द बेसलाइन इज हियर, राइट? यू कैन सी दिस डॉट. इफ इट वाज सेट देयर इट मे हैव गिवन मोर क्लीन डेटा कंपेयर टू दिस वन. दिस वाज ऑटोमेटिकली सेट बाय मशीन, है ना? तो समटाइम्स द सॉफ्टवेयर फेल्स. इन दैट केस यू हैव टू सी इट. टू improve the quality of the data you can manually also set the baseline, right? So first method is the quantitation, is the absolute quantification which is used for the measurement of the exact copies of the nucleic acid, gene copy number and virus load estimations. So this is we are going to perform in the practicals. And how we can do that? We compare, we first we generate a standard curve by comparing the CT value of the known quantities of the DNA template. Then the unknown sample CT value is plotted or analyzed with the standard curve to get the quantity of the DNA in that sample. So these blue dots are the test samples or unknown samples, है ना? तो इस रेड सैंपल्स इट इज स्टैंडर्ड कर्व जनरेटेड बाय कंपेयरिंग द नॉन क्वांटिटी ऑफ डीएनए यूजिंग qPCR. So qPCR gave a CT value for all these templates, early CT for higher quantities, less CT mean greater CT for less quantity. So when we compare the CT value and known quantities we get a standard curve. So when we regress this CT value and the known quantity DNA template, we get a linear regression equations which is y is equal to mx plus b. Using these equations you can draw simple these equations to calculate the copies of DNA in your test sample manually. You can do it in Excel. If you properly label your standard and samples in qPCR, the machine or algorithm will automatically do it for you. We used to use step 1 plus qPCR from applied biosystem that is having. I don't know whether this Bio-Rad wala has or not. So we will see. If we are not getting it in machine itself, then we will do it in manually in Excel, right? Like वो आप HPLC, if you have done it HPLC or other like spectrophotometric quantification, you might have already performed this, है ना? तो हाउ टू जनरेट, द क्वेश्चन अराइजेस हाउ टू जनरेट ए स्टैंडर्ड कर्व. आप वो नॉन क्वांटिटीज लाएंगे किधर से? बट यू हैव सीन इट लाइक इन PCR, वॉट एवर द प्रोडक्ट ऑफ द प्रीवियस साइकिल कैन एक्ट एज ए टेम्पलेट फॉर द नेक्स्ट साइकिल. सो PCR प्रोडक्ट वी कैन यूज इट. क्या यूज़ कर सकते हैं हम लोग स्टैंडर्ड कर्व जनरेट करने के लिए? PCR हमने लिया, उसको प्योरीफाई किया, देन स्पेक्ट्रोफोटोमीटर से उसका क्वांटिटी लिया. Now I know this length. Now मुझे पता है कि मेरे PCR प्रोडक्ट की लेंथ क्या है ना? एंड क्वांटिटी क्या है, मैं नैनो ड्रॉप से मेजर कर लिया. बेस्ड ऑन क्वांटिटी एंड लेंथ आई कैन कैलकुलेट द कॉपी नंबर्स यूजिंग वन कांस्टेंट कॉल्ड एज Avogadro constant, this one. And there are freely online software where you can calculate the copy number of DNA, है ना, वो भी कर सकते हैं. तो आपने PCR प्रोडक्ट लिया, उसको उसमें कॉपीज पता किया, उसको सीरियल डाइल्यूट किया, देन यू कैन रन on qPCR. Now qPCR will give a CT value for each known samples. You develop a standard curve and run उनको कर लो. ऐसा कर सकते हैं. Can we do it? कर सकते हैं. Can we do it? Yes or no? Just can we do it or no? Yes or just no? थोड़ा आवाज़ बोलो, मैं सुन नहीं पा रहा हूँ. हैं? थोड़ा जोर से बोलो, मैं सुना ही नहीं. कर सकते हैं, नहीं कर सकते? कर सकते हैं, but you won't get as good as this standard. You won't get it, and every time you will get a different standard, because PCR product में all PCR products are not equally synthesized. मतलब वो कुछ मैच होगा उधर और भी बेकार चीज़ें होगा, मतलब हाफ होगा कैसे भी करके. That is not the clean one. So how to get a clean standard curve? Just take an example and understand this one. I want to estimate the TOLC NDV in a white fly. So what I will do, and my primers for qPCR are, this is the CP gene, are somewhere from middle में से है CP gene से for qPCR. So I will take another primer pair which will amplify my entire CP gene. वो है, उसका साइज है 771 बेस पेयर. मैं उसको एम्पलीफाई करूँगा. देन आई विल क्लोन दिस प्रोडक्ट इन ए सूटेबल वेक्टर. टीए क्लोनिंग के साथ मैं कर लूँगा, मैं इसको क्लोन करूँगा. सो व्हाट विल हैपन इन टीए क्लोनिंग, एक पीसीआर प्रोडक्ट ना, एक सिंगल पीसीआर प्रोडक्ट, इट विल गो एंड लाइगेट विद ए प्लाज्मिड. सो वन ई कोलाई सेल्स में वन रिकॉम्बिनेंट प्लाज्मिड जाएगा. नाउ वी कॉल इट रिकॉम्बिनेंट प्लाज्मिड. एक सेल में एक ही जाएगा. Now we isolate the plasmid, recombinant plasmid from the E. coli. Now check whether your gene of interest is there or not, that in plasmid. So go for a restriction digestion. So I perform a restriction digestion. Now you can see my PCR product will be there. इसके साइड में भी these are the restriction site, इसके साइड में भी these are the restriction site, दोनों साइड में है. So अगर मैं if I cut it from both side, I will get a my gene of interest there on gel, right? If I cut it from one side, the plasmid will get linearized. वो पूरा circular में नहीं रहेगा, it will get linearized. So that gives a good estimation. आप circular से standard बनाने की कोशिश करेंगे, that also will not give a like reliable standard every time. वो आपको every time अलग अलग PCR efficiency के साथ में PCR होगा. So you have to cut it from one side. Then you have to run on gel. अभी लीनियर होगा, you won't see this band, but you will see only this band. Now you have to go for gel purification, not PCR product purification, because अभी भी you isolated the plasmid from the E. coli, still that may be having some amount of nuclear DNA. वो nuclear DNA होगा, उसमें हमारा gene of interest नहीं है. When you go for quantification, वो nuclear DNA will give थोड़ा बहुत प्रॉब्लम क्रिएट करेगा. सो गो फॉर जेल प्यूरिफिकेशन. तो यहाँ पे पूरा प्लाज्मिड होगा, प्योर प्लाज्मिड, वो भी एक पीसीआर प्रोडक्ट का, एक सिंगल डीएनए स्ट्रैंड का, रिकॉम्बिनेंट प्लाज्मिड के साथ में. ओके, क्लियर अप टू दिस? सो अभी मेरे पास एक ट्यूब है जिसके अंदर मेरा एक रिकॉम्बिनेंट प्लाज्मिड है. उसको मैंने लीनियराइज कर दिया है by cutting it using a restriction enzyme from a single site. Now I go for a spectrophotometer or nanodrop based reading. Last time I told this is the 771 base pair long PCR product, and this is 3.9 kb my plasmid. So based on the total length and quantity in that tube in maybe nanogram, we can calculate the copy number in that tube. Now I know my first tube is having 10 की power 7 copies of DNA. Now I can dilute it. I have to dilute it 10-fold serially, maybe up to 10 की power 2 copies or 1 copy. So 10-fold serial dilution से हम लोग क्या कर सकते हैं? Now my each tube is, in each tube I know what is the concentration of the DNA. Now you know it exactly, right? Clear है? So when you know these quantities and you run in a PCR product, it will give a CT value, है ना? You can see it clearly. अगर लेटर CT है इसका मतलब DNA क्वांटिटी कम है, अर्ली CT इसका मतलब क्वांटिटी ज़्यादा है, और वो you have to get this equal spacing because you diluted it properly, right? 10-fold serial dilution है तो 10-fold serial dilution का स्पेस मिलना चाहिए इस एम्प्लीफिकेशन में, है ना? Then you get this standard curve. अभी तक क्लियर है? So now you have the standard curve. अभी क्या किया आपने? You have isolated DNA because TOLC antibody is a DNA virus. You need to isolate DNA. Tomato leaf curl nudity virus is a DNA virus. So from whitefly I will isolate the DNA to quantify the load of the virus, है ना? So this is my standard. This is the plate layout of the real time 96 well plate. So I will load my standard in each replication. You can see, if you can see, it is 10 की power 7, right? It is 10 की power 7. It is 10 की power 6, 5, 4, 3, 2. You load the standard curve and load the unknown samples. So unknown samples will give some CT value. The machine algorithm will automatically calculate the quantity of DNA in each sample. You can see it, if you can clearly see it. इधर दिख रहा है आपको थोड़ा प्लेट में? दिख रहा है? नहीं दिख रहा, थोड़ा छोटा है, कोई बात नहीं, ठीक है. So we will see in practical. So every time when you are going for quantification, every time आपको ये standard प्लेट में लोड करना पड़ेगा. ऐसा नहीं कि एक बार आपने ये स्टैंडर्ड कैलकुलेट कर लिया, आपने ये अननोन सैंपल रन किए, जो CT वैल्यू मिली एक्सेल में आपने कैलकुलेट कर दिया. थोड़ा वेरी करेगा, थोड़ा रिजल्ट ऑलवेज वेरी करेगा. टू गेट इट वेरी क्लीन, डू इट एवरी टाइम. अभी व्हाट थिंग्स यू हैव टू टेक केयर व्हेन यू आर डूइंग क्वांटिटेशन यूजिंग द एब्सोल्यूट मेथड. योर अननोन सैंपल मस्ट बी विद इन द रेंज, बिकॉज आपका स्टैंडर्ड लीनियर रहेगा आउटसाइड दिस रेंज कोई गारंटी नहीं है. सो योर अननोन सैंपल मस्ट बी विद इन द रेंज ऑफ दिस स्टैंडर्ड. अगर उसमें क्वांटिटी ज़्यादा है, डाइल्यूटेड, लेट्स से आपका ये इधर ज़्यादा क्वांटिटी वाला ना, आपके सैंपल में क्वांटिटी बाहर जा रही है इधर, वो अभी जाएगी, मैं थोड़ा चेक किया हूँ, तो वो इट मस्ट बी विद इन द रेंज ऑफ द स्टैंडर्ड. Or you have to run the standard every time when you are quantifying the unknown samples. And the PCR efficiency always matter. So the PCR efficiency also software will automatically tell you based on the standard, this slope of the standard. This particular standard is having efficiency of 100%. 100% mean my PCR product is doubling at each cycle. After each cycle they are getting exactly double. Mean my enzyme is very efficient and my primers are very specific, so they are going and binding to the specific DNA, not here and there. So efficiency, it must be 100%. Now another thing is the slope, it must be 3.3. If efficiency is 100%, it will be 3.3. The efficiency range can be acceptable from 90 to 110. If your efficiency is less than 100%, it means your consumables are old or your primers are not proper. They are having some kind of primer dimer or non-specific binding. if your efficiency is greater than 100%, it means you are doing some mistakes, you are doing pipetting error. ठीक है? And so we have to talk about the primers ना. Primers must be specific. It should not have primer dimer or non-specific binding. If you are getting it, वो कैसे पता लगेगा आपको? मेल्ट करो एनालिसिस से. That too in cyber green chemistry में. टैकमैन में तो नहीं होगा, but टैकमैन is very costly. You can't afford every time. So मेल्ट करो एनालिसिस से you will come to know whether your primers are specific or not. अगर स्पेसिफिक नहीं है सो डाइल्यूट इट. प्राइमर्स को आप डाइल्यूट करेंगे तब भी आपका प्राइमर डाइमर का प्रॉब्लम्स इट कैन बी मिनिमाइज्ड बट कैन नॉट बी एलिमिनेटेड. इफ यू आर स्टिल गेटिंग ए नॉट प्रॉपर मेल्ट कर्व देन रीडिजाइन योर प्राइमर्स. देयर इज नो वे टू रिमूव दैट. एंड व्हेन यू आर डेवलपिंग ए स्टैंडर्ड कर्व ऑलवेज यूज ए कैलिब्रेटेड पाइपेट्स. वो आपका कैलिब्रेटेड पाइपेट होना चाहिए, है ना. नहीं तो एवरी टाइम इट विल गिव सम डिफरेंट वॉल्यूम्स, सो यू वोंट गेट ए प्रॉपर स्टैंडर्ड कर्व. हाउ गुड यू आर इन pipetting और setting reactions, you will exactly know with the qPCR, because a single mistake will be shown here. You will see with your eyes how much mistakes you are doing in pipetting and other things. So use larger volumes for serial dilutions in standard curve. अगर आप कम वॉल्यूम यूज़ करेंगे always you will see some errors in pipetting. अगर small error है pipetting में then you will see a big difference over here. तो larger volume for better precision. आप स्टैंडर्ड कर रहे हैं तो आप एटलीस्ट 100 माइक्रोलीटर तो बनाइए ही बनाइए. उससे ज़्यादा बनाएंगे that is more better. Another was relative one. So it is not there in the practicals. This is used for gene expression studies. Relative, a term itself is saying relative, mean compared to someone, right? And who is that someone? A calibrator, a control sample. So let's take an example. Who is the calibrator and who is a control sample? Let's say I have two varieties. One is the Kufri Jyoti and Kufri Bahar. Kufri Bahar is known for virus resistance against TOLC and DV. वो पहले ही पता कर लिया था हमने कि हाँ ये it is showing less symptoms and it is good one, it is showing some good response against the TOLC and DV. So I want to check the Kufri Jyoti also, whether this is also, mean it may be more samples, but I want to check Kufri Jyoti whether this Jyoti is resistance to TOLC, mean not resistance, it means how much it can tolerate the multiplication of virus, है ना. So I can categorize in different resistance categories. So I want to check whether this Kufri Jyoti is also resistance or not. So I will go for a relative quantification of virus gene expression after infection, right? So I will take Kufri Bahar as a calibrator. So I will compare the multiplication of the virus, a gene of virus inside this Kufri Jyoti at some time periods, right? So the 3R of Kufri Bahar will act as a calibrator. Compared to 3R, I will see the expression in fold change at all these points, Compared to this point, this is called as calibrated sample. Compared to, the name itself is telling the relative. In relation to the Kufri Bahar, what is the fold change of the virus gene expression? So for multiplications, actually we take a replicate gene because it will replicate inside. So it will go there, it will make a mRNA and proteins, है ना? So we are measuring the mRNA level in this variety and in this variety. So this is my calibrator sample, है ना? So another one is that we are comparing these two varieties. So we need to take exactly same amount of RNA to compare, right? Comparison तो तभी होगा जब आप equally opportunities दे रहे हैं, है ना? So exactly we take the equal amount of RNA to convert them in cDNA, then seeing the gene expressions. So that is really impossible. there is no way to measure exactly same amount of RNA in variety A and variety B. There is no way. आप स्पेक्ट्रोफोटोमीटर से भी 2 बार रीडिंग लेंगे, यू विल गेट 2 डिफरेंट रीडिंग्स, राइट. नैनोड्रॉप किसने यूज किया है, आपने देखा होगा. इफ यू डू द सेम सैंपल अगेन यू विल गेट द डिफरेंट रीडिंग, राइट. देयर इज नो वे एग्जैक्टली यू कैन मेजर. सो व्हाट इज द वे आउट टू नॉर्मलाइज दिस? वी यूज एंडोजीनस कंट्रोल्स. Endogenous controls are housekeeping genes whose expression is constant with any stimuli. आप you give any stimuli, उनका expression it will remain constant. अगर आप वायरस का इन्फेक्शन भी दिया है तो उस जीन को कोई मतलब नहीं है, वो अपना काम कर रहा है, वो उसका एक्सप्रेशन कांस्टेंट है. सो यू हैव टू चेक इट. वो ऑलरेडी पब्लिश्ड है जीन, तो कोई बात नहीं कि हाँ ये एंडोजेनस कंट्रोल लिया जा सकता है वायरस के केस में. So you use that. अगर वो नहीं है then you take 4-5 endogenous control housekeeping genes, give the infection and check whose expression is constant. उसको हम लोग endogenous control use करेंगे. So let's say you took थोड़ा बहुत slightly difference है, error है हमारा पाइपेटिंग में, हमने exactly same RNA नहीं ले पाए, तो उसको सेम DNA भी सेम इफिशिएंसी से नहीं बना पाए, देन यू आर गोइंग फॉर जीन एक्सप्रेशन, तो थोड़ा बहुत वेरिएशन आएगा. देन हाउ टू नॉर्मलाइज, बिकॉज वो जीन का एक्सप्रेशन तो कांस्टेंट है दोनों वेराइटीज में, है ना. अगर पाइपेटिंग एरर हुआ है तो उस जीन का भी एक्सप्रेशन में थोड़ा बहुत डिफरेंस मिलेगा, उतना ही डिफरेंस मिलेगा जितना आपका पाइपेटिंग एरर हुआ है, ठीक है. सो टू नॉर्मलाइज दिस यूनिट मास यू यूज ए एंडोजेनस कंट्रोल. सो आपको 2 चीज़ें चाहिए कि वन इज कैलिब्रेटेड सैंपल जिससे आप कंपेयर करेंगे जीन एक्सप्रेशन के केस में कि इसके कंपेयर में इतना फोल्ड डिफरेंस है इस जीन में. दूसरा यू नीड ए हाउसकीपिंग जीन जिसके कंपैरिजन में आप जीन एक्सप्रेशन को नॉर्मलाइज करेंगे, ठीक है. सो यू गेट द CT वैल्यूज फॉर योर कंट्रोल सैंपल. देयर इज द कुफरी बहार इज द कंट्रोल सैंपल एंड ट्रीटमेंट इज द कुफरी ज्योति 1, एंड यू गेट the CT value for housekeeping gene and gene of interest. Gene of interest is the replicate gene of that virus, and housekeeping gene you can't take it from the virus, you have to take it from the plant, ठीक है. तो प्लांट का कोई हाउसकीपिंग जीन है, उसका CT वैल्यू ले लिया आपने और जीन ऑफ इंटरेस्ट का CT वैल्यू आ गया. बेस्ड ऑन CT वैल्यू यू कैलकुलेट द डेटा सेंसिटिविटी डेल्टा CT which is the gene CT minus the housekeeping gene CT. You can do it in the Excel itself. There are so many videos there in YouTube also, you can see that one and clearly you can do it. After you getting a delta CT, we calculate a delta CT value which is the sample delta CT minus the control delta CT, and to know the fold difference of these gene expressions you raise this delta CT 2 की power minus delta CT. So you get how much fold difference is there. It may be in negative, like Kufri Bahar may be having expression of some gene higher than the Kufri Jyoti. It may be negative, it may be positive. So you'll see the significant difference is there, at least 2-fold difference is there. It is the significant difference. It has to be more than 2-fold difference. मतलब उसका expression 2 गुना तो होना ही होना चाहिए. So that's it for virus estimations using qPCR. I hope you got some clarity. अभी कोई doubts है तो you can ask. Thank you. Is there any question you may ask? हाँ, sure.
So this housekeeping genes, we will be standardizing it based on like multiple experiments.
Hmm. Mm-hmm.
But like if you are checking gene expression some different type of treatment other than this where the housekeeping gene is already checked,
Mm-hmm.
can I use the reference gene from here or do I have to check the housekeeping genes again for my experiment?
It is a good idea to check it again, but still if you are not if you don't mean like if you are lacking with consumables another time, then you can use it, no problem. In that case use 2 housekeeping genes, है ना. So the mean of 2 housekeeping gene will definitely normalize mean chances of your like error. So use 2 housekeeping gene in that case. If you are not aware the stability or constant expression of that housekeeping gene in that different experiment, then use 2 housekeeping genes.
How much variation we can like ignore like in housekeeping genes? For example,
You
here
it has to be minimum. It has to be minimum. Let's say you perform let's say you gave infection and you are checking the expression of the 5 endogenous controls. Which one is the least one? Use that one. There is no, I think there is no exact demarcation that is safe for immunization.
like between treated and control we have here 17.1 and 17.0.
अच्छा, it may be it is a good idea to actually ना वो जो आपका हाउसकीपिंग जीन है ना उसका CT वैल्यू बहुत लेट भी नहीं होना चाहिए. It should not be more abundant also. आपके जीन का CT वैल्यू 20 या 30 नहीं होना चाहिए. ऐसा नहीं कि अपन जीन कंट्रोल का 30 से ऊपर आ रहा है तो आपका वो मेन कॉम्पोनेंट में होना चाहिए.
Wide. Wide. That's fun.
Then you are then you are going to be upregulated or so if you do that then तो you will use like 1.5 and yours the same what it is a linear and it will be like you can see on paper it is published in this is all the first author in the paper and you can follow exactly standard also you can use the primers and same following the paper, right? We are not doing the I can like it again and I can send you back. Right? If you are doing, you may ask, मतलब हम लोग क्या what primers we are using for qPCR? And we find the gene of interest using normal PCR, clone it, and follow the same procedure and go for it. What is can we do that? Primers range में normal PCR जो भी प्रोडक्ट आएगा उसको क्लोन करो go for it and can we do it or not? थोड़ा big थोड़ा fast हो yes or no? No yes है mostly yes exactly we can do it what will happen is exactly the same but standard को दे दीजिए I will design my own primers and go for it then so वो it doesn't matter so like my CPG you can understand you don't have to go for cloning ये वो ठीक है
What is the case for RNA viruses? Can we do instead of DNA isolating? इसमें तो CDA or you go for a normal media. You can say what the difference, the fold difference in sample A and sample B. What is the fold difference? Exactly how much fold is there? Go for relative production. The fold difference only. Numbers. If you don't have a control these forces, somebody क्या करना पड़ता है? I didn't allow मतलब वो वायरस नेगेटिव है you need you certain में then तो आप relative quantification you can you can't compare. There is no way to compare, na? You need some expression. Then anyone आपका है you don't have yours then you have अच्छा जी then it will be like अच्छा we can also discuss like active dye you will be using is तो उस मास्क में one more dye who knows what you said correctly who I use to reduce license to what cyber make it give some warning to cyber then it will affect your results definitely when it goes to the DNA we give the light then it will not give fluorescence then why light both dyes are light but one is interconnected one is not rocks को process बाहर आएगा तो भी same process let's say you made basket and you distribute it create mass mix cell, you are distributing it. It is not that cell. It gives some different प्रोटीन चला गया how to है ना, तो वो 14 देगा प्रोसेस तो वो पूरे बेस का नॉर्मलाइज करता है पिपेटिंग एरर को दिखती है प्रो ठीक है any idea of this stage is not a pathology. Which one will transmit? Instead of force here, this stage of transmit. So because of this body, only the process are mixed from the virus. After it goes to the certificate gland. Through the certificate gland, when the certificate is there, then it actually transmits. So all these the basic of the virus binding. And also that quantification of genomic tools for insect diagnostics. There is some lecture because of some periods. After this lecture, we have two interesting lectures. One by National Biotechnology, Dr. Ramcharan Bhattacharya will be delivering. This lecture will be on then another lecture will be our joint diagram as well. So two interesting lectures are lined up. First is about this lecture, and thank you, Dr. Shaka, for delivering this lecture. I can take it from Chula. Small moments of you. Explaining and ICAR.
Wait, sir, wait. Sir, sir. Wait. Yeah. ठीक है सर, थैंक यू.
Any clarification I will clarify because after reaching that and so that's why we give it to the if there are additional inputs you can send it.